All right. So today's lecture will be shifting away from what we've been doing previously. So up to this point, we've been talking a lot about molecular biology and how we get proteins, the different classes of proteins, how we can measure those proteins. We're going to pivot now to a little bit more of a cell bio perspective, particularly on as it relates to immunology. And we'll be talking about that within the context of Crohn's disease. Crohn's disease falls under the category of being a type of inflammatory bowel disease. There's two different kinds of inflammatory bowel diseases. There's Crohn's disease and there's ulcerative colitis, or just sometimes just dubbed colitis. The way in which they differ is what segments of your GI tract they affect, and the way in which it manifests within where the effect on those areas with those lesions. So ulcerative colitis, as the name implies, typically is restricted to the colon, and it affects typically the mucosal layer within the colon. Within Crohn's disease, it can go from anywhere from the mouth to the anus, and it typically has lesions that go all the way across the intestinal wall. There's also a further characterization of what arm of the immune system or what cells of the immune system are typically associated with those diseases. We're going to talk more about those cells in our next lecture, but to give you to kind of give you a little bit of information now, the ulcerative colitis is typically a Th2 mediated disease, whereas Crohn's disease is primarily Th1 and Th17. And those are different flavors of T cells, and T cells are a big cell population within adaptive immunity. And our next lecture is going to be all about adaptive immunity. So you'll understand more about what a Th1 is, a Th2, Th17, or just a T cell in general. But for now, we're going to focus on a separate arm of the immune system. The symptoms associated with Crohn's disease, because of the effect on the ability for Crohn's disease to manifest across the entire GI tract, this can really disrupt nutrient uptake and manifest with growth retardation, with fever, and with severe weight loss as well. The way in which this manifests on the GI tract is that there are these abscesses on the intestinal wall, and these abscesses are these pus-filled lesions. They're like these sites of severe infection, and it's pretty uncomfortable. It's pretty painful. For those of you who've had an abscess before, bless you. Imagine a lot of times people get them on their digits or on their limbs, but having it internally on your GI tract is incredibly painful. It also can lead to these ulcers, these basically holes within the intestinal wall that eventually can lead to something that's called a fistula. A fistula is typically a hole that's created between two cavities or two open areas. And one common way in this can occur is either a fistula between the rectum and the bladder, or between the rectum and adjacent nearby cavities. And so this is incredibly painful, incredibly problematic, and is a way for these pus-filled abscesses to spread infection. One thing I forgot to note is that the abscess, and whenever you see pus, that's typically a certain cell of your immune system called the neutrophil. That's kind of like the first cell to come in when there's some sort of infection, and the pus is a bunch of just dead neutrophils that are there. And so that's typically whenever you see pus, it's just a bunch of dead cells. In addition to the effect on another consequence of that nutrient uptake deficiency is people with Crohn's disease typically have anemia. In addition to that, they also have neurological issues as well. The prevalence is pretty high. There's about 1.4 million people alive with IBD. So that's either colitis or Crohn's disease. And the prevalence can also be pretty high as well. There's 1 to 10 cases per 100,000 people in the United States. There's typically a bimodal distribution. That means if you were to graph the number of cases of IBD as a function of age, there would be two peaks where people between ages of 10 to 20 would be one peak, and people above 50 would be another peak. Why is there bimodal distribution? I don't really know, but there's because there are two different arms of inflammatory bowel disease of Crohn's and colitis, there could be some explanation as to where one presents at an earlier age and another one presents primarily at an older age. Yes.
So is the 1.4 million figures just for Crohn's disease or IBD?
IBD.
And for an incidence, what's the distribution by?
I don't know. We talked before about disparities among autoimmune diseases. To my knowledge, because this is not an autoimmune disease, I don't know of any gender or racial disparities as it relates to IBD, but that's actually a good question. I'm going to look into that because I don't know of any, but that doesn't mean that doesn't exist. Yeah, that's a good question. All right, so the etiology. So the causes tied to Crohn's disease. We talked before about monogenic diseases. We talked about sickle cell disease. We briefly talked about cystic fibrosis as another monogenic disease. Crohn's disease is one where there's multiple genetic mutations that seem to enhance patients' susceptibility to developing it. And so because there's multiple genes associated with it, we would identify Crohn's disease as being a polygenic disease. There are multiple genes associated with it. And the mutations seem to have a common theme of causing the innate immune system to malfunction and not work properly. That's going to be the arm of the immune system we talk about today. And because of that dysfunction in innate immunology among these patients, Crohn's seems to develop when there's an overcompensation from the adaptive side, the other arm of the immune system, that seems to promote the emergence of Crohn's disease. And there's also the belief that the symptoms that typically emerge among patients with Crohn's disease, that it's also in response to constant immune responses to our microbiota or microbial infections within the GI tract. What's worth noting, what we're going to focus on today, is that there's a specific gene that's associated with Crohn's disease called NOD2, and that is a critical gene. And it's also a critical receptor that's associated with innate immunity. And so we're going to come back and bring everything back to Crohn's disease when we get to NOD2. So to this point, I've talked about innate and adaptive immunity. And so there are two arms to our innate immune system. And the way in which our immune cells develop is something, they emerge from these stem cells within our bone marrow called hematopoietic stem cells. This is not something I'm going to quiz you on, in that I'm not asking you to memorize. The process of these stem cells becoming immune cells is called hematopoiesis. I would not ask any of you to memorize all of the cells involved with hematopoiesis. Most grad students don't, so I'm not going to ask you guys to do that. But the reason why I bring this up is because all immune cells emerge from these hematopoietic stem cells, but they kind of bridge off into the adaptive immune cells, primarily the T and B cells, as well as the innate immune cells over here, the myeloid progenitors. And so a lot of the cells that we're going to talk about are on this left side here, on this slide, that come from this common myeloid progenitor. And so these are all of the different actors within our innate immune system on the left side here. This was talked about in the pre-lecture, but it's worth repeating, but it's also important to note because these qualities of the immune response and qualities of our immune system will differ. It's going to be true of both arms of the immune system for both innate and adaptive immunity, but how they are true will differ. And so the way in which one of the first key qualities of our immune system is that there's an incredible diversity, and there's an incredible ability to respond to a wide array of pathogens. For our innate immune system, there's a specific set of pathogens that our innate immune system is able to respond to, and part of that's because it recognizes specific qualities or patterns or features on microbes or on cells that are not supposed to be there. So if you remember, we talked about early on in this class that our immune system is designed, its main purpose is to distinguish between what is self and what is foreign. And the specificity here for innate immunity is that it's able to recognize and respond to specific patterns and features on foreign elements that are not supposed to be there. And there's incredible memory associated with our immune system, and particularly, at least, with our innate immunity. This memory is genetically encoded in our innate immune cells. What that means is, when we typically think of our immune response or our immune system, we think of typically getting a vaccine and our body remembering whatever that antigen is or whatever that virus is so that we can respond to it appropriately. That's true for adaptive immunity. For innate immunity, the memory typically that we think about is the ability to pass down genetically encoded receptors that respond to these specific features or patterns on foreign microbes or foreign elements. There's also a really cool growing area of research right now called innate memory. We typically think of innate memory, again, as like genetically encoded receptors that respond to pathogens that are present across shellfish, rodents, all orders of mammals that have been genetically passed down and encoded. But there's also a unique factor, innate memory, where it seems like cells are able to see some sort of pathogen, and there's some sort of genetic change also that enables a pass down of memory. That's an active area of research. That's really cool. So that's not the quality of innate memory that I want you guys to take home, but rather to try and give you a piece of information of where the field seems to be going, seems to be trying to identify now. So we have our innate immune system and our adaptive immune system. We can further kind of break down our immune systems to two qualities: our humoral immune response and our cell-mediated response. There's typically, when we talk about the humoral immune response, we typically mean what the factors or features of our immune system that show up in our blood or in other bodily fluids. And so for innate immunity, the innate humoral response is something called the complement system, where it's a series of enzymes that are meant to go in and directly kill some sort of foreign cell or some sort of foreign pathogen. The cell-mediated innate immune response here is typically by cells engulfing pathogens through phagocytosis. We've talked about phagocytosis before. It's the process of cells eating or engulfing some sort of foreign pathogen or some sort of particle, or in the context of immune response, we think of it as some sort of foreign pathogen. It can also be eating up and clearing up unnecessary particles or debris that shouldn't be in that local area. And so, again, innate humoral immune response is the complement system, these series of enzymes that we'll talk about on the next slide that are meant to go in and destroy a pathogen. The innate cell-mediated response is typically phagocytosis. On the adaptive side, the adaptive humoral response are antibodies. These antibodies that are able to inactivate and neutralize some sort of foreign pathogen and help us kind of convey that memory, that long-lasting memory. And there's also the cell-mediated adaptive response, where these cells respond to pathogen-infected host cells. So like virally infected cells are basically undergo apoptosis or are destroyed by specific cells of the adaptive immune response when they can identify a virally infected cell. Before I jump into the complement system on the next slide, I want to give space for a couple questions here. Yes. Yeah, so typically, we're going to talk about this a little bit later, but or we're going to go in greater detail about this next class. But typically, whenever there's some sort of viral infection, I'll take one step back. Every cell in your body has a protein on it that says, "I belong here. I am." So like I have cells that say like, "I am Dan," and there's like a specific protein on them that convey. It's almost like an ID card that say they belong here. A virally infected cell will not express that protein properly, and in response to some sort of viral infection, there's going to be proteins that are expressed that tell every other cell, hey, really express that ID protein on you so that I know who is healthy and who is not. And so typically the virally infected cells won't express that ID protein properly. And that's how we can target specifically those cells who seem to have some sort of, they're virally infected and their machinery is being hijacked so the virus can replicate. That's what the virus is trying to do. Like a virus's goal is to hijack our machinery so it can replicate. And when there's some semblance of a virally infected cell, every cell begins to express that ID protein. It's called MHC class 1. All of them express that protein to an even greater degree so that our immune cells can go in and survey and find the virally infected cells. Yes?
Could you give the exact definition of the innate versus adaptive immunity?
Yeah, so the exact definition is that innate immunity. I'll give you a layman's description of it that I've always loved, and then I'll give you a more precise definition. Innate immunity is fast but dumb. It happens really quickly on the order of hours to a few days. And it's dumb because it only recognizes patterns or features on microbes and pathogens. It doesn't like remember something for 10 years, like one single epitope that it's destined to respond to. It just recognizes, it has receptors where it recognizes specific patterns on microbes and pathogens. Our adaptive immunity is slow, but incredibly smart and elegant. So it takes a while, it takes longer for your adaptive immunity to kick in. But once it does, you have antibodies that are produced by your antibody producing cells, B cells, that last you for years. And so most of you, if, you know, there's a lot of people who got vaccines when they were kids. If you were to go to your doctor and see those titer levels, you'd probably still have antibodies for Hep B, for MMR. So it's fast and elegant because it has specific cells that are supposed to respond to specific epitopes. Their entire life, we have cells in our body that are meant, T cells and B cells are meant to respond to a specific single epitope. That's their entire destiny. And so once they see that epitope, they replicate. So then we have our B cells are making antibodies that respond to that epitope. If it's a chickenpox or if it's MMR, or not MMR, that's a combination of things. If it's Hep B, there's a specific T cell and a specific B cell that's destined to respond to that. So it takes longer to get there to ramp up those cells. But once they're there, and once you have activated those cells, you have protection for a long, long time. And so the adaptive and innate immunity are defined by those roles, as well as defined by who those actors are. Adaptive immunity is typically T cells and B cells. It's kind of reductive to say it this way, but typically T cells and B cells. And innate immunity has a lot more players in it. We have our neutrophils, our macrophages, our mast cells, eosinophils, basophils. So like these are all cells on that left half of that hematopoiesis slide. In case everyone downloaded the slides early, great job. I updated them today to include a couple new slides. So in case you're not seeing them on your set of slides, I just updated them earlier today. And so that's typically how we define innate versus adaptive immunity, how it functions, as well as who the players are contributing to those functions. Yes.
So, assuming you have been infected by a particular pathogen before, it's fair to say that the innate immune system is set up to buy turns for the adaptive immune system.
Not by time. So like typically it's like the first line of defense. So like if there's some sort of pathogen that has a pattern on its surface that a macrophage can respond to, it can get eliminated that way. If it has some sort of, if we already have, or if it has some other feature that our cells can respond to, or if it tries to enter into our cells and it has a quality that our other parts of our innate immune cells can respond to, it'll be eliminated that way. If it evades detection by our innate immune system, then what begins to happen is there's a specific cell that's kind of the bridge between innate and adaptive. It'll break it down, present it, and go to our lymph nodes and say, okay, are there any T cells that recognize this? And then there are B cells that are going to look at it and say, like, okay, that's my epitope. That's what I'm meant to respond to. And then adaptive immunity will take over and kick in.
So I'm assuming the name is that those specific features that you recognize are just common features to say common on most of all bacteria.
Hold that thought. Yes, we're gonna talk about that. We're gonna talk about what some of those features are. But yes, on specific kinds of bacteria.
And if so, why is it not just
Because we've developed protection against them.
right? So they should be like eliminated because the ones who don't have those features wouldn't survive, but those who do would successfully invade and replicate.
Good. That's a good question. They're not all. So not to create a hierarchy, humans are a higher order mammal. Not all mammals have the same exact immune system. So what humans have as far as innate immunity is not what a mouse has, is not what other lower order organisms have. And so that enables them to continue thriving despite our immune system providing protection against them. I'm going to keep moving in the interest of time. So the complement system. The complement system is a fairly complex system of lots of surface proteins. To focus on what the take-home message here is, this is the humoral component of our innate immune system, and the key function of complement is to try and destroy pathogens that complement is designed to respond to. There are typically three pathways that are associated with the complement system. We have the classical pathway, we have the lectin pathway, and we have the alternative pathway. I'm not going to ask you to memorize every single step in each pathway, but there's going to be commonalities across these pathways that I do want you to focus on and know. And I also want you to know how each of these pathways are initiated. So know the overlap of these pathways as well as how each of them is initiated. First, what we're going to do is we're going to focus on the classical pathway, which is on the top left of this image here. Typically, what will happen is that on some sort of bacterial pathogen, that we're going to have antibodies that are going to recognize specific features on the surface of this bacteria. They could be the same antibodies that recognize the same epitopes on this antigen, on this bacteria, or they could be recognized. They could be two different antibodies that are recognizing two different epitopes that are on the surface here. But what's key to know is that classical pathway is initiated by a complement protein called C1. It almost looks like a spaceship that comes in and like docks on the surface of the moon. It comes in and binds to these antibodies that are on the surface of this bacterium. And what begins to happen is that this C1 protein begins, once it's there, it'll see another complement protein called C2, and it'll cleave it into C2A and C2B. From there, we'll have another complement protein called C4, and C4 will be cleaved to C4A and C4B. What's key to note here is that C2A and C2B come together to form an enzyme complex. It's called the C3 convertase. All right. So again, we have C1 that comes in and binds to these antibodies on the surface. And typically what you'll see is most of the time you'll see C2 and C2, C3, C4, like all these first set of complement proteins. This is an alphanumeric soup here, but again, focus on the big picture, the overlap, and how these things are initiated. Classical pathway has these antibodies that bind. This C1 protein comes in and docks on those antibodies. It cleaves C2 into C2A and C2B, and then it'll also cleave C4 into C4A and C4B. C2A and C4B make an enzyme together. That's called the C3 convertase. I'm going to hit pause right there on the classical pathway. All right. So know that this is the we're gonna stop on the formation of this enzyme called the C3 convertase, that is C2A and C4B. Now, the lectin pathway is initiated by this similar, almost spaceship-looking like protein that docks on the surface of a bacterium. That complex is called mannose binding lectin. Mannose is what I'm going to do is I'm going to create like a virtual asterisk to try and note ways in which our cells are different from some pathogens or bacterium, and how when our immune system leverages those differences to recognize those pathogens. So typically on the surfaces of our cells, mannose is a kind of sugar. We don't typically have a lot of mannose on the surface of our cells. Bacteria have a lot of mannose on their cells. So our immune system over generations or over many generations, what's happened here is that this protein, our immune system has developed a protein here that leverages that difference and has this lectin protein that binds to mannose-rich surfaces, like on the surface of bacterium. And so mannose-binding lectin is what docks and binds to these mannose-rich sites on a bacterial cell wall. And then that will cleave C2 into C2A and C2B. It'll cleave C4 into C4A and C4B. And we get the C3 convertase, C2A and C4B. Okay? So both of these pathways converge on making this C3 convertase. That C3 convertase will clip C3 into C3A and C3B. And so the easy way to remember this is those two complement proteins that come together, C2A and C4B, they make the C3 convertase. It's an enzyme. In the name is its substrate, what it's targeting. The C3 convertase targets C3. And so C3 is clipped into C3A and C3B. And typically what happens is C3A is an anaphylatoxin. What that means is it's a component that is that induces anaphylaxis. There's typically when there's an anaphylactic reaction, we think of that in the context of allergy. But what ends up happening is that there are local immune cells in that area that are activated, that have receptors for C3A, that have receptors for C3A, that then will release histamine that cause edema and typically fluid loss from the vasculature into the local area. And so you'll typically have an anaphylactic reaction. There's typically anaphylaxis associated with C3A because it's an anaphylatoxin, forgive me. This alternative pathway, what ends up happening is that as C3 is being cleaved, a lot of these bacteria have something on it called endotoxin. It's typically like a feature or a pattern on the surface of these cells that will bind to C3b. And so all of these pathways will converge on C3b being clipped and being present here on the surface of the cell. So just to repeat, what are the key take-home points here? The classical pathway begins with antibodies binding to the surface and the C1 protein docking. Mannose-binding lectin pathway, the lectin pathway initiates with this mannose-binding lectin protein complex that docks and binds to mannose because that's a unique feature that's highly expressed on the surface of bacterial cell walls. And they converge at making this C3 convertase, this enzyme that cleaves C3 into C3A and C3B. C3b stays there, and C3A is an anaphylatoxin. For the alternative pathway, typically C3b from these other pathways typically will bind to endotoxin and be wedged in the surface there. And so that's another way to activate another complement pathway. All of these pathways are not mutually exclusive. You might have the classical pathway and alternative pathway happening together, or the lectin pathway and the alternative pathway come together. And so to this point, what we have is we have C3b present on the surface of this bacterium. What ends up happening is you end up getting once you add C5b, forgive me, once you add C3b, it becomes C5 convertase and it clips C5. And once that happens, you add C6, C7, C8, it makes this huge complex. And now what I actually think this is pretty cool. What ends up happening is that once you have that complex with all of these complement proteins, C9 comes in and it starts drilling a hole in the surface of that bacterium. And that disrupts the integrity of that bacterium cell wall. And we call it the MAC complex, the membrane attack complex. And it destroys the pathogen. And so this is happening all over the surface of this pathogen. And the goal is to destroy it. And this is the way it does it. And so a few key main outcomes, takeaway here, a few key main outcomes to focus on for complement is that we get these enzymatic byproducts that are anaphylatoxins, C3A and C5A. what will end up happening is that it'll prompt the blood vessels, again, like edema. Our blood vessels get leaky and there's fluid loss from the blood vessels into the local area. That makes it easy. The blood vessels are leaky there now. It's easy for immune cells in our bloodstream to go to that local site because it's leaky there. So it recruits phagocytes. The C3b that's coded on the surface, whether it's from the classical pathway, the lectin pathway, or alternative, that C3b is actually becomes a target for phagocytes to engulf and undergo phagocytosis of whatever that pathogen is that has C3b. The final outcome, another alternative outcome, is this MAC complex that destroys the pathogen. I'm going to stop for a couple of questions there. Yes.
So where do complement proteins go to?
In our bloodstream.
So if they're in the bloodstream, but it requires an activation to, I guess, cause the immune to get the bloodstream, how does it?
So that's a great question. Typically, what ends up happening whenever there's some sort of injury or infection, there are these like inflammatory proteins that are expressed that also causes some degree of edema or inflammation. Like when we talk about inflammation, a lot of times in immunology we talk about like specific proteins that are expressed. There's also just like classic inflammation where like if you get a bruise, if you have an injury, there's inflammation there, and things swell up, things get red, things get painful. What's happening is that the inflammation is causing immune cells to get recruited there. Our blood vessels are leaky. Lots of proteins and cells are going to that location, and it's painful because that edema is making things swell up and impinging nerves. So any sort of infection or some sort of pathogen is going to involve some sort of inflammatory response that's going to involve edema or cause our blood vessels to get leaky. And when it's leaky, those complement proteins are going to be secreted to that area so that it might respond in case there is some sort of pathogen that they can target. Does that make sense? That's a good question. Yes.
Could you explain the alternative pathway that is C3 from the other pathways end up coating the surface of bacteria?
Yeah, so what ends up happening is that the C3b can do one of two things. The C3b can either, on the second bullet point on this slide, can become a signal for phagocytes to engulf this pathogen, or can help promote the creation of the next enzyme that targets C5, called the C5 convertase. Yes.
So is the mechanism like the grand finale for all of these pathways, or just for those?
For all of these pathways? That's a great question. The complement, its grand finale, like you said, is the MAC complex. Along the way, these other things can happen where phagocytes are recruited, and if they see C3b, they can engulf and destroy the pathogen. But if that doesn't happen, then complement can go all the way and be completed with the formation of this MAC complex to destroy the pathogen. It does. Well, it helps lead to its destruction because the anaphylaxis, again, a key part of it is that it's promoting our blood vessels to get leaky. And so it's making it easier for phagocytes to be recruited to that site and engulf and undergo phagocytosis there for those pathogens. I'm going to do two more questions, then we're going to keep moving.
So for the second half, you're pretty much, it's identical to the plasma pathway, except you skip the C1 cleaving, you go straight to C2 and C4b.
You can envision the difference between the lectin pathway and the classical pathway is that instead of C1 and binding to these antibodies on the surface, you just have mannose binding lectin docking there, binding to mannose rich regions and leading to the cleaving of C2 and C4.
Gotcha. and then that forms C3A, like C3, and that causes cell lysis. But the result of the classical lectin pathways is that you have the enzymatic byproducts.
Mhm.
And then for the alternative pathway, you have with the 3B.
You have C3b that can either be serving as a signal for phagocytes to engulf it, or helping serve as more binding sites to create that C5 convertase, that C5 enzyme that helps continue on with the next steps here.
They all finish with that.
Yes. Last question.
The C byproducts are just all consumed by a phagocyte anyways, right? They're not recycled or
Sorry, the specific, sorry, it actually, not, let's go
what's
say
the MAC? What happens?
those will likely get, what's good, that's a good question. What happens to complement proteins after pathogens have been destroyed? Some of them might get engulfed. So if this pathogen is being engulfed by a phagocyte, they're not going to be recruited. They're going to be destroyed. If this pathogen has been destroyed by the MAC complex, the debris will be engulfed by phagocyte. So they're not going to be they're not going to be recycled. We have specific cells that express these complement proteins that will replenish the expression of these complement proteins after they've been cleared out, for lack of a better phrase.
So what happens to the other components of C4 and C2 after the C2A and C4B when they little complex?
That's a good question. They're not in aflatoxins like C3A and C5A. That's a good question. I actually don't know. I'll look into that, but I don't know off the top of my head. In the interest of time, I'm going to press on because there's some key stuff I want to try and get to. So we're going to try and now look at cell-mediated innate immunity. I talked before how there's specific patterns or features that our cells are going to recognize. And these patterns or features are called pathogen-associated molecular patterns or damage-associated molecular patterns. That's a mouthful, so we're going to call them either PAMPs or DAMPs. And they are recognized by pattern recognition receptors. What's really interesting is that these PAMPs, these pathogen-associated molecular patterns, they're these unique features that are these like repetitive structures that are on the surface of pathogens that have been targeted by innate immunity over the course of evolution. So over the course of evolution, these PAMPs have been identified in that there are receptors, these pattern recognition receptors that have been expressed that recognize these PAMPs. Similarly with DAMPs, you can think of DAMPs as, because it's damage associated, they're almost like a flare gun whenever there's some sort of tissue repair problem that goes on. And so both PAMPs and DAMPs are recognized by pattern recognition receptors and are key features of our innate immune system and its ability to induce a cell-mediated response. Some of these DAMPs can be ATP, uric acid, or even sometimes cytokines. But what's unique and what's noteworthy is that these are typically things that are intracellular that are not meant to be extracellular typically. So like typically ATP is like within a cell. It's part of the energetics of the cell that causes reactions to occur. If there's extracellular ATP available, that typically means there's a cell that's been ripped apart because of some sort of tissue damage event. Same thing with specific cytokines, not all of them. And cytokines, those are just soluble proteins that serve as cell-to-cell factors, like signaling factors between cells. And the pattern recognition receptors are, again, these host-encoded proteins that recognize PAMPs and DAMPs to trigger an innate immune response from cells. Some of these pattern recognition receptors will recognize factors that promote chemotaxis. Chemotaxis is chemical-mediated movement. Cells are migrating towards some sort of chemoattractant. And so there are some pattern recognition receptors that'll induce chemotaxis for it to move and follow some sort of pathogen. On the pre-lecture, there was a really cool figure or a really cool video of a neutrophil just kind of going in and following around a pathogen. And that was involving the engagement of a pattern recognition receptor that was driving that neutrophil to move and to undergo chemotaxis. Movement to follow that attractant. There are anti-pathogen defenses where we have pattern recognition receptors that, again, recognize specific patterns on pathogens that it'll respond to and induce an innate immune response. This will trigger the release of cytokines. Again, these are soluble proteins that are kind of like signaling cues between cells. Historically, we only think of them as being among immune cells. We're also starting to see now that there are a lot of different cells that express them, that communicate to immune cells and induce certain reactions. There are also chemokines. Chemokines induce chemotaxis. You can think of them as another soluble factor, but this time, instead of simply just being a signaling cue, it is promoting movement of the cell, migration, chemotaxis. And finally, it promotes changes in gene expression. And so these pattern recognition receptors are a key feature of cell-mediated innate immunity. And one last key thing that I'm going to illustrate for you all is that these pattern recognition receptors can be extracellular, or intracellular, inside the cell. If you think about it, if a virus successfully infects and enters into one of our host cells, we would want a defense mechanism in case that virus gets into our cells and starts hijacking our machinery to replicate and make more of itself. And so we have pattern recognition receptors intracellularly and extracellularly. One classic pattern recognition receptor is called formal peptide receptor, or FPR. It's a family of G protein coupled receptors that recognizes specifically formulated peptides. What's noteworthy is that whenever a protein is synthesized during translation, there's usually something called a start codon that triggers the synthesis of that protein. It's called AUG. We're going to talk about what codons are later, but you can just think of it as a sequence that triggers the synthesis of a new protein. And it typically encodes for the amino acid. It begins with encoding methionine as the first amino acid for any of our proteins being made. Again, this is virtual asterisk of like how we are different from pathogens. In many bacteria, their methionine is formulated. Ours is not. So that is one way in which our immune system has identified how our peptides are different than peptides of a pathogen. And so the formal peptide receptor binds to formulated methionine, leucine, and phenylalanine. And the key part, the asterisk here, is that it's a formulated methionine, because our methionines are not formulated. Pathogens typically are, bacteria specifically. And so this triggers, when it binds to the FPR, that triggers a really strong chemotactic response from neutrophils and macrophages. And typically, we only encode for 3 different FPRs, and mice actually encode for more. So I talked earlier about how typically higher order organisms like us have a more robust immune system than lower order organisms, not to denigrate mice or other mammals. But there's an evolutionary response why mice would have more FPRs that respond to bacteria than we do. Does anyone want to venture a guess what would be the explanation for that? Yeah? Sorry? I wouldn't say they have a faster immune response. I would think about, sorry, go ahead. That's exactly right. They are filthier than we are, for lack of a better phrase. And so they need an immune response that responds to so many different kinds of bacteria. And so that's typically like one like evolutionary response or evolutionary explanation as to why mice, in this case, have more FPRs than we do. Another class of pattern recognition receptors that are kind of like the I don't want to say main ones, because different immunologists have their thing that they really like. But whenever we talk about pattern recognition receptors, typically the ones that come to most people's mind are called Toelike receptors. These are located both intracellularly and extracellularly. They're called Toelike because typically in a lot of immunology, some of the first discoveries were made in flies. That's a pretty common and easier to work with model system. And Toll is kind of if I remember, I think a Latin word for fly. But when the same receptors were identified in humans, we just said that they are Toelike receptors because these Toll receptors were first found in flies. We have 10 different Toll-like receptors that have been identified. We don't know everything about each of them. And so again, I said this to someone at a recent office hours, and I meant to say this on the first lecture, I may not have. This is a class of like half-truths, where like there's a lot we know, but there's a lot we still don't know. And that's particularly true with immunology. Some of the cells that have been identified were identified in 1905 or 1910, where someone for their PhD like discovered a cell, which is kind of weird to think about. But there's still so much we're learning about immunology, which is why some of these things have a question mark on them. And there was even someone here at UVA during their graduate work that kind of identified what TLR9 binds to and what it really triggers. And so the ways in which these Toll-like receptors, the ligands that they bind to, are associated with where they're located. So for example, double-stranded RNA is something you would we have single-stranded RNA. A virus typically has double-stranded RNA. You would typically see that double-stranded RNA when there's a viral infection and that they're taking over our machinery to replicate. And so we would want something inside our cells in case there's double-stranded RNA around. And so TLR3 is intracellular. Flagellin is recognized by TLR5. That's on the bacteria. That's something that would be extracellular. LPS is lipopolysaccharide. That's a feature on the surface of a bacteria. And so and same thing with LTA, lipitoic acid. Those are both, that's TLR2 and TLR4, expressed extracellularly. CPG, so those are regions of DNA that are rich in C's and G's. And so there's a difference between ours and theirs, but we would only see a bacteria's DNA if it's been engulfed and is available intracellularly. And so TLR9 is expressed intracellularly. And so this is a quick illustration of where the different Toll-like receptors are expressed. And particularly TLR3, 7, and 9 are usually expressed along with an endosome where these engulfed pathogens are being destroyed, and where we would see where they would see their respective ligands. So one of those ligands that we talked about is lipopolysaccharide. It is particularly present on gram-negative bacteria. It is the key feature of what's on gram-negative bacteria, and we're going to show why that is. It typically has a glycan polymer on the top, a core oligosaccharide, and it's also known as endotoxin. So when we talk about, or when you hear about endotoxin, LPS is what's being discussed, and it's such a potent activator of our immune cells that whenever something's being injected or implanted, it needs to be confirmed that it is endotoxin free, because even the smallest amounts can trigger a really strong immune response. And so for different bacteria that are defined as being gram-negative and gram-positive, that's in relation to the gram stain being able to bind to peptidoglycan. So for gram-negative bacteria, they have these two membranes, an outer membrane that has lipopolysaccharide LPS on the surface. There's this periplasmic space with the peptidoglycan and another membrane. So because this peptidoglycan that binds to the gram stain is not accessible, this is gram-negative. Gram-positive bacteria only have this one membrane with peptidoglycan on the surface and another feature, another lipoprotein that we'll talk about in a moment that's present that identifies them. But we'd call these gram-positive because the gram stain can bind to the peptidoglycan that's on the outer surface of these bacteria. And for these gram-positive bacteria, they have something that's called lipitoic acid present that is either wedged within or through the peptidoglycan. And so it has a DAG anchor that anchors it to the one cell membrane that gram-positive bacteria have. But again, so LPS is a key feature of gram-negative bacteria, and lipitoic acid is a feature of gram-positive bacteria. Based on the time, we are going to stop there. I was hoping to get through all of it, but we're going to pick up where we left off, and hope you guys have a good spring break.